So today we will understand Unit 3 in Advanced Enzymology. The Unit 3 includes enzyme mechanism and its regulations. Enzyme mechanism and its regulation. To understand that, one should understand how to measure or determine the mechanism through which enzyme substrate complex is worked. What are the rules of regulators in the metabolism? Then what are the bifunctional enzymes, pseudoenzyme, enzyme promiscuity, then extremoenzyme, catalytic nucleic acids, or you can say ribozymes. So we will understand these 3.1 to 3.6. So before going to that, enzyme mechanism. What is enzyme? You know very well. Enzyme includes all enzymes are protein, but all proteins are not enzymes. Enzyme composed of certain specific site, active site, as well as enzyme bind with the substrate. It will convert into ES complex, and finally it will convert into a product. So similarly, these reaction can be possible on, if we see the enzyme, as you know that it is a biological catalyst and universally all the reaction can facilitate with it. So typically it is a protein, and these protein do not involved in that remain as such and enhance the rate of reaction. So speed up the rate of reaction by lowering the activation energy. Enzymes are highly specific, that I already given a certain note enzyme specificity and their types also. So it can catalyze wider range of reactions. That already been taught to you. What is active site, how many active site are present in on the enzyme, and how enzyme can regulate. So enzyme have a region at which you will find the active site, which is like a small pockets where the molecules can bind, and this active site has a specific site for various chemical properties that already been taught to you, how it is inhibited and how it react with also that allow interact to substrate and facilitate the reaction, and finally it gives into a perform that reactions. There are one theory I have already explained, induced fit model and lock and key models. So in similar to these lock and key models, specificity of enzyme substrate interactions that have a specific shape to bind it with a specific substrate. Similarly, you can see in the key fit with the lock. In induced fit models that undergo conformational changes upon the binding site. And these models already been explained to you why some enzymes can catalyze multiple reactions. That is the answer. This model allows for greater flexibility. Therefore, this induced fit model is suitable for that. ES complex mechanism of enzyme mechanism include these enzyme plus substrate that will gives you an enzyme substrate complex. Optimally position of these substrate and that conversion of substrate into product can only be possible with the K1 and K2 that I will explain in the Michaelis Menten equation also. So catalytic mechanism. Now start the topic. Enzyme enjoy various catalytic mechanism. Various catalytic mechanism. For what? To facilitate any biological reactions. Some common mechanism includes. Now you can write. Some common mechanism include acid-base catalysis, covalent catalysis, metal ion catalysis, and proximity or orientation effect. Now tell me loudly.
Acid base catalysis, covalent catalysis, metal ion catalysis, and proximity or orientation effect.
Yes, these mechanism involve plus proton transfer,
Protein transfer.
formation, or a breakage of covalent bond, and metal ion coordinations, and bringing reactant into a close proximity. These are the respectively you can see with this reaction. Now read what is catalytic mechanism of enzyme, how it can explain in a simple word.
Enzymes employ various catalytic mechanisms to facilitate reactions. Some common mechanisms include acid-base catalysis, covalent catalysis, metal ion catalysis, and proximity orientation effect. These mechanisms involve proton transfer, formation or breakage of covalent bond, metal ion coordinations, and bringing reaction into close proximity, respectively.
So seventh point, I am explaining one reaction rate and enzyme kinetics. These chapters I will explain in depth with the Michaelis-Menten equation. So enzyme kinetics is the study reaction rate and enzyme kinetics. Enzyme kinetics is the now right. Enzyme kinetics. If it is asked in the examination, how to write that definition? Enzyme kinetics is the study of the rate at which enzyme catalyze reaction only. The rate at which,
Activate enzyme catalyze reactions.
but study of.
Study
Study
of
of. The reaction rate depend upon various factors that I already explained in the factor affecting enzyme activity. So substrate concentration, enzyme concentration, temperature, pH, presence of inhibitor, presence of activators. Enzyme kinetics can be described, explained with the mathematical models. And there are two equations. One is Michaelis-Menten equation, another is Line-Weaver-Burke. So this is all about the reaction rate and enzyme kinetics. Enzyme regulations. So enzyme regulation, there are various ways I can explain with the six inhibitions. First, allosteric regulation, write down. Covalent modifications, feedback inhibition, competitive inhibitions, then non-competitive inhibition, enzyme activations, and itself enzyme inhibitions. So these enzyme regulations, first allosteric regulation. Allow. Allosteric regulation occurs when molecules bind to a site on the enzyme, and that site called allosteric site, that is distinct from the active site. Not that site, as I say, allosteric site, it compete with that. So allosteric regulation occurs when the molecules bind to a site on the enzymes that is distinct site from the active site. This binding can either enhance or inhibit the enzyme activities. Certainly, we are only understand allosteric regulation that enhance the rate of reaction. When enzymes comes, you understand it is the study of mechanism of the rate of enzyme reactions. But here allosteric site at that can be the other than the active side, and the binding can either enhance or activity, that may be a positive regulation or a negative regulations. So this regulation, if it is positive regulation, then you can call it as positive allosteric regulation. If it is the negative regulation, then it is called negative allosteric regulation. The enzyme activity, allosteric regulation can small molecules or other proteins or it is made of proteins. So in short, if it is asked in the examination, what is allosteric regulation, you have to write like this. Now another is covalent modifications. So covalent modification involves the addition or removal of a chemical group to an enzyme, which can alter its activity. So example of covalent modification include the photophosphorylation or no phosphorylations, acetylation, methylation, and glycosylations. What are the example of covalent modification?
And examples for phosphorylation,
Very good.
acetylation, methylation, glycosylation.
Yes. These modification either active or deactivate the enzymes.
Enzymes.
That is also, as I said in the previous allosteric regulation, it
Enhance or inhibit the chemical
so
enzymes.
it can inhibit or enhance.
Huh.
So they have two types of regulation. One is positive, another is negative regulation. Here is also same thing is that. But here you will not find that only activate or deactivate. By what? It involves the addition or removal of chemical groups to the enzymes and which can alter its the specific activity. So example of covalent modification, as I said, it is include acetylation, methylation, glycolization, or phosphorylation. Now feedback inhibition. I will give you an example with every differences between allosteric regulations and covalent modification with example. Then feedback inhibitions. Feedback means I have already explained in intermediary metabolism also. There are certain enzyme are key regulators. Okay, phosphofructokinase. Similarly, feedback inhibitions. That feedback inhibition is a regulatory mechanism in which the end product of metabolic pathways inhibits the earlier enzymes in the pathway. So these are maintain the homeostasis or prevent excess
Access.
production of particular metabolites. So feedback inhibition is every time necessary for formation of whether it is from lactic acid or a pyruvic acid. If the similar mechanism is continuous in the glycolysis or gluconeogenesis, what will happen? That will create a certain problems. So feedback inhibitions is a regulatory mechanisms in which the end product is suffered or metabolic pathways can be inhibited with the earlier enzymes in the pathways. So itself enzyme is a feedback inhibitors. So particular product is or excess production of metabolite is inhibited, that is called feedback inhibitions. So competitive inhibitors or non-competitive inhibitors, non-competitive inhibitions means that is also molecules that is resemble to the substrate and complete for the binding of active site of an enzyme. So if it is asked in the examination, you have to write competitive inhibitors are molecules that resemble the substrate and complete for the binding. Compete, not complete. Compete for the binding to the active site of an enzyme. They reduces the enzyme activity by the preventing the substrate from the binding effectively. So now up to this we have completed four. First, allosteric inhibition, then
Chemicals, chem,
covalent modification, then third,
feedback inhibition or
competitive inhibition.
uh,
Hmm. competitive inhibition, feedback inhibition, but before that covalent modifications and previously
Illustrated.
allosteric. Now non-competitive inhibition. So that binds to a site of an enzyme other than the active sites. So you can say the allosteric site and inhibits activity that by causing a conformational changes that affect the substrate binding or a catalysis. Enzyme activations, that is also one of the regulators. So enzyme can be activated by the various mechanisms such as binding of activators, removal of inhibitors, and activation can occur through the conformational changes. It includes all, as I discussed previously, that enhance the enzyme catalytic activity. Enzyme inhibition refers to the reduction or complete reductions or a complete cessation or complete closing of the enzyme activity. So inhibition can be a reversible or irreversible. So enzyme inhibitions can be classified into two, and one is reversible inhibition, another is irreversible inhibition, depending upon the nature of the inhibitors and the interactions with the enzymes. So this is all about I have explained uh, enzyme mechanisms. Enzyme mechanisms, and then these chapters includes enzyme mechanism as well as enzyme regulation or regulators. So until that you have to explain if it is asked in the examination 1 to 7, right?